Hello students, welcome to another lecture in Understanding Biology chapter 31, The Living Plant. In this lecture we will talk about phloem transportation, plant nutrition and plant hormones. At the end of this lecture you should be able to describe the process of translocation, explain how sugars move through the plant body using the pressure flow model of phloem transport, distinguish between macronutrients and micronutrients, describe the properties of plant hormones including auxin, cytokinins, gibberellin, ethylene, and abscisic acid, and how they affect the plant growth. As leaves produce sugar through photosynthesis, plants need to transport these products to other parts of the plant, such as root for storage and all the other parts for energy. As we now know that transportation in xylem is unidirectional, but the phloem transportation is bidirectional, can happen in both directions. And the process of phloem transport is known as translocation, and not only sugars and sucrose is transported by phloem, they also transport hormone, mRNA, and other molecules including variety of sugars, amino acids, organic acids, proteins, and ions. So when it comes to translocation, most widely accepted model explaining the sugar moving through the phloem is called pressure flow model. So dissolved carbohydrates flow from source to a sink. Carbohydrate sources are where photosynthesis is happening, such as mesophiles of leaves. The food storage tissues, on the other hand, such as cortex of roots, can be either sources or sinks. Sinks are highly metabolically active tissues. Let's look at how sugars enter into the phloem at the source. This loading process is known as phloem loading. When sugars are generated in mesophil cells via photosynthesis, they enter the phloem via smallest veins. Some sugars travel from mesophil cells to companion cells and CU cells via symplast. But much of the sucrose are loaded to CU cells via apoplastic transport and is moved via a sucrose and hydrogen symport. This is achieved by proton pumps. So if you remember, we talked about sieve tube elements. They are actually not alive at maturity. So they need companion cells or parenchyma cells to provide ATP for this transportation. But this bulk flow can happen without any further energy requirement after this sugar is loaded into the sieve tube elements. So this is because after it is loaded to the sieve tube elements, they use principle of water potential to move this material in the phloem through the phloem system. So the water potential in sieve tube elements are actually higher water potential compared to xylem. Therefore, water flows into sieve tube elements by osmosis. The turgor pressure actually increase in these sieve tubes, and this pressure drives the fluids through the plant system of sieve tubes. At sinks, the sucrose and hormones are actively removed from sieve tubes and water flows by osmosis. At sinks, therefore, turgor pressure drops. So what you need to remember is that how sugars are loaded into the sieve tube elements, because they are not alive. They need ATP generated by their companion cells or parenchyma cells. Once they are loaded into the sieve tube elements by these proton pumps which require ATP, the rest of the movement can happen without requiring any energy, because they use principle of water potential. Compared to water potential in sieve tubes, xylem cells have higher water potential because xylem have less iron concentration and phloem has more sucrose and hormones and other stuff, so their water potential is less. Therefore, water actually flows into the sieve tube from the xylem by osmosis. This will actually increase the turgo pressure in the sieve tube elements, and then this drives the fluids through the plant's system of sieve tubes, and when it goes to the sink where they had to unload the bulk, what happens is that sucrose will leave these sieve tube elements and the turgo pressure will actually go down, and then at the sink they actively remove this sucrose and the water will actually go out by osmosis afterward. So here is a diagram of the mass flow of phloem. They are actually bidirectional, as you can see both ways, the arrows, and at shoot tip, the sink, the water flows out passively into the xylem, where they are actually going to give the sucrose out at the shoot. That is why it is a sink, and the sucrose is actually actively transport out, and turgo pressure will drop down and water will move out. So here is another picture here at a leaf, which is a source. Now they are uploading the sucrose into the sieve tube elements, and this is actually energy requiring process that need the help of companion cells. Once the sucrose is loaded up into the sieve tube elements, their water potential drop down compared to the xylem, so water will move into the phloem, and this will drive the move of the phloem contents in the phloem to a sink somewhere. It could be root, it could be a growing portion. So that is how mass flow is happening in the phloem. So I hope now you have a good idea of how movement happen in the phloem. So now that we talked about that, let's move on to plant nutrition. So what kind of nutrition is required for plants to grow healthy? So we already know that plants require carbon dioxide, water, and light energy for their photosynthesis. Photosynthesis is a major physiological process that happen in plants that provide energy to the plant to grow. Apart from these, there are actually large quantity of inorganic nutrients that required by plants for their growth and well-being. So these nutrients can be divided into two categories. They can be macronutrients that required in large quantities, and others that required in minute quantities are called micronutrients. So we have macronutrients and micronutrients. There are 9 macronutrients such as carbon, oxygen, hydrogen, nitrogen, potassium, calcium, magnesium, phosphorus, and sulfur. And then we have 7 micronutrients including chlorine, iron, manganese, zinc, boron, copper, and molybdenum. So any of these nutrients, any deficiency can lead to have severe effects on plants. As you can see in this picture, compared to healthy tomato plant, magnesium deficiency, manganese deficiency, and copper deficiency can give drastic variation in color and the healthiness of the tomato leaf here. So those are the nutrition requirement of plants. Now we know that how plants are transporting water and nutrients and what are essential nutrients, but majority of other day-to-day life activities of plants are actually relying on complex physiological networks. Many of these internal signaling pathways depend on plant hormones. In this section, we are going to look at several important plant hormones. So what are hormones? Hormones are actually information containing chemical substances produced in small quantities in one part of the organism and then transported to another part to give a response. In animals, most hormones are produced at a specific site, but in plants, hormones are not produced in specific sites. There are 7 major kinds of plant hormones. These include auxin, cytokinins, gibberellin, brassinosteroids, oligosaccharides, ethylene, and abscisic acid. But in this lecture, we are only going to discuss about auxin, cytokinins, gibberellins, ethylene, and abscisic acids. So let's first look into auxin. So discovery of auxin is pretty interesting. Charles Darwin himself actually studied plants. He and his son Francis Darwin reported that growing plants respond to light through phototropism. He hypothesized that when seedlings are illuminated from one side, they bend towards the light in response to an influence transmitted downward from the source at the tip of the shoot. 30 years later, Peter Boysen-Jensen and Arpad Pal showed that the substance causing the shoot to bend is diffusible chemical. They did the agar block assay, where they cut the tip of a seedling and place it on top of an agar block, and the chemical diffused to the agar give the same effect that is placed on another topless seedling. In 1926, First took a step further with the agar block experiment, and he found out that a diffusible substance in the tip of the light grown seedlings can make seedlings bend. So this bending is actually due to signaling molecule causing the cells on the side of the agar block to elongate more than on the other side of the seedling. He named this chemical as auxin, and auxin promoted faster elongation on the side where the light is. That helps seedlings to bend towards the light. So here is first Wendt's experiment. So he did he took these oat seedlings and put them, illuminate them into the light, and then after that he cut off the tip of these seedlings and placed them on a agar, which is a gelatinous substance. So whatever the chemical that is produced on the tip of this shoot, because of the illumination of light, will diffuse into the agar block. So once it's diffused, he has these dark grown seedlings. He placed this block of agar in the side of these seedlings. As you can see that when you place them in the side that exposed into this agar block has bended now. So even though these were not exposed to the light, they still bended because of that chemical that diffused from that agar block that was exposed to the light. So that was his experiment. So this chemical enhanced rather than retarded the cell elongation. So cell elongation was enhanced, and first Wendt named these substances as he discovered auxin. So auxin acts to promote growth and elongation. Environmental signals can influence the distribution of auxin in the plant, especially light. Light can affect the production, distribution, and destruction of auxin in plant, and auxin actually promote the activity of vascular cambium and vascular tissues. One of the direct effects of auxin is an increase in the plasticity of the plant cell wall. This is only in younger cell walls, lacking the secondary cell wall formation. And further, the acid growth hypothesis provide a model linking auxin to cell wall expansion. According to this hypothesis, auxin cause responsive cells to actively transport hydrogen ions from the cytoplasm into the cell wall spaces, which decrease the pH. These activate enzymes that break the bonds between cell wall fibers, allowing the cell wall to expand. So even though auxin has been researched extensively, auxin's molecular basis of action is difficult to figure out yet. Unlike animal hormones, a specific signal is not sent to the specific cell. Most likely multiple auxin receptors sites are present in plants. Auxin is also unique among the plant hormones because it is transported towards the base of the plant. Now that we are done talking about auxin, let's move and look at another plant hormone, which is called cytokinins. Cytokinin in combination with auxin can stimulate cell division and differentiation. Most cytokinins are produced in the root apical meristems and transported throughout the plant. You can find cytokinin in developing fruits. Cytokinin actually promote the growth of lateral buds into branches. Cytokinin inhibit the formation of lateral roots, where auxin promote the lateral root formation. Therefore, balance of auxin and cytokinin is very important for balanced growth of plants. The next hormone we are going to talk about is called gibberellin, and gibberellin are actually named after the fungus Gibberella fujikuri. This fungus grows parasitically on rice plants and causes them to grow abnormally tall. Gibberellin belong to a class of more than 100 plant hormones. All are acidic. Therefore, gibberellin is abbreviated as gibberellic acid. This hormone have important effects on stem elongation. This stem elongation effect is enhanced when auxin is present. Another hormone we are going to talk about is called ethylene. Ethylene is a gaseous hydrocarbon and was known to defoliate plants for a long time. Even before we understood that this is actually a plant hormone, ethylene was used in these lamps, and wherever these kerosene lamps are located, the plant actually shed their leaves. So we have been knowing about ethylene for a long time. Ethylene is naturally produced in small quantities and interact with other plant hormones to regulate plant growth and development. When auxin is transported downward from shoot apical meristem, it stimulates the production of ethylene in the tissues around the lateral buds, and these prevent their growth. Ethylene is also suppress the stem and root elongation, but ethylene actually plays a major role in fruit development. At first, auxin stimulates the ethylene production, which will fasten the fruit ripening. The last hormone we will discuss today is called abscisic acid, which is synthesized mainly in mature green leaves, fruits, and root caps. Application of abscisic acid can stimulate fruit dropping in cotton. Abscisic acid induces the formation of winter buds that are dormant and remain through the winter, and it also plays a role in seed dormancy. Another important fact about abscisic acid that it is actually involved in controlling the opening and closing the stomata. We did talk about it earlier. Also, it is very important to induce dormancy and prevent premature germination called viviparisy. So viviparisy happen in strawberry, you can see in this picture. So abscisic acid is very important to prevent this premature germination. So in summary, Translocation is the movement of dissolved material through phloem in plants. Phloem transportation occur according to the pressure flow model. Carbohydrates loaded into sieve tubes create difference in water potential. As a result, water enters the tube, creating a pressure to move fluids through the phloem. Plants require 9 macro and 7 micronutrients. Hormones are chemicals produced in small quantities in one region of the plant and then transported to another to exert a response. I hope you enjoy this lecture. Until we meet with another, have a good day. Thank you.